Good evening, everyone. Welcome to today's session, Basics of Clinical Research, Approach and Opportunities. I'm Smriti Venkatesh, the Administrative Director at the Global Surg Network. With me, we have the founder and president of GSN, Akatya Vidushi, our PRC Director, Reena Mehta, and Social Media Director, Sneha Yathali. The Global Surg Network is a student-led surgical society which aims to bridge the reach of global surgery through surgical education and training. Welcome to our session, Basics of Clinical Research. I would like to thank all of you for joining us today. It is my honor to introduce our esteemed guest speaker for today's session, Dr. Sabyasachi Sengupta. Dr. Sengupta is the founder and director of Future Vision Eye Care and Research Center, Mumbai. He is also the founder and director of Sengupta's Research Academy, a comprehensive portal providing online lectures on research methodology and services to assist manuscript preparation. He is the current associate editor of the Indian Journal of Ophthalmology. Sir has published 90 plus articles in peer-reviewed journals, presented more than 25 papers and posters in national and international conferences, and won many prestigious awards for his outstanding achievements. We are all very excited to hear from you, sir. Before we begin with the session, I would like to establish a few ground rules to be followed at all times during the event. We request all of you to keep your mics off. Feel free to keep your video cameras on during the event. In case you have any doubts, please put them in the chat box and they will be addressed towards the end of the session. Also, the session will be recorded and uploaded later on our YouTube channel. Without further ado, let's begin with today's session. Over to you, sir.
A very good evening. Thanks, Smriti, and thanks to the Global Surgical Network for giving me this opportunity to be here and interact with you. So it's always exciting to interact with young minds, impressionable minds that you are now, and the world is really at your feet. You can do so many things, and I'll try and show you some of these opportunities that and how you can approach them. I'll just quickly share my screen. And let me know when you can see my screen. All right, so I think my screen is visible.
Yes, sir, we can see it.
All right, so I always like to start with a bit of inspiration, and here, so I keep changing this with the kind of audience that we have. So I think for you, I think it's important that you ask questions. So the questions you ask define who you are, and not the answers you know, right? Because you are still evolving and you're young, and it's time for you to ask questions. So I think it's important that you keep this in mind. So we'll look about talk a little bit about clinical research, certain approaches and steps that you can follow, and the opportunities that are out there for you. And I think we'll concentrate more on opportunities because the approach is relatively standard. So the roadmap to doing research and finally publishing really is this. So you plan a study or your research, and you execute it, you analyze and write it up, and finally you review the paper and you submit to a journal, and finally it gets published. So this is generally how we do it. I've already been introduced, so this is me, and I've had specific training in clinical research techniques at the Johns Hopkins School of Public Health and the Johns Hopkins School of Medicine. That's the Wilmer Eye Institute. I'm an ophthalmologist, and I have about 100 publications, and I have assisted more than 100 publications through my research academy. And also I'm the associate editor of the Indian Journal of Ophthalmology, right? So that is the flagship journal that we have in ophthalmology today. And so I think from a writer's point of view, and then from a mentor's point of view, and now from an editorial point of view, I think I can give you some unique perspectives on the opportunities that you will have, and the approaches that you can take. I think this is a clip that is going to come up next. I think it has it really has inspired me for more than a decade now. So just take a quick look at this.
दिलों में तुम अपनी बेताबियां लेके चल रहे हो तो जिंदा हो, नजर में ख्वाबों की बिजलियां लेके चल रहे हो तो जिंदा हो, हवा के झोंकों के जैसे आजाद रहना सीखो। तुम्हें एक दरिया के जैसे लहरों में बहना सीखो। हर एक लम्हे से तुम मिलो, खोले अपनी बाहें। हर एक पल एक नया समा देखी है निगाहें। जो अपनी आंखों में हैरानियां लेके चल रहे हो तो जिंदा हो तुम। दिलों में तुम अपनी बेताबियां लेके चल रहे हो तो जिंदा हो तुम।
I'll just let that sink in a bit. For me, this is what clinical research really means. I think it gives me freedom, it gives me opportunities, and my heart really yearns to do more and more of this. So, I hope some of you will, have these feelings, as in when you evolve in medicine. Before we dive into the nitty-gritties of all this, I thought that what I want to quickly discuss with all of you is, how will medical practice look like when you are actually in practice, or when you are actually into mainstream practice. So how will medical practice look like in 2030? So I want you to quickly think about this in your own mind, and have maybe one or two ideas in your mind before I show you how I think medical practice will look like. I think in one word, it is going to be automations. Automations are going to revolutionize the way everything is going to be practiced. And a lot of the processes that we are doing today in patient care, in diagnostic, in therapeutics is all going to be automated. So, I tell a lot of students. I was talking to students from BJ Medical College in Pune, I think sometime last year, and I was telling them the same thing. Don't put all your baskets, all your eggs in one basket. That's because, what we learn today might be entirely automated tomorrow. Then what are you going to do? So you need to be multifaceted. You need to develop many, many skills other than just patient care. On your left, these are the 5 levels of automations that have been frequently described. Initially, it was only humans. Then there was a shadow mode where some amount of computerization was happening. We today are at the AI assistance phase. So if you look at the images on the right, so this is an image of the retina. And these are some pathologies, and if you look at the image on the bottom, you will see all those blue dots. That's basically a machine which is a software which is scavenging that image for differences in pixels, and it is picking up all those images, right? And it is giving us an actual diagnosis. So you don't really need a human to actually diagnose that there is something wrong here. So we are in the phase of AI assistance, but we are going quickly towards automations, and there might be full automations very soon, right? So I think this may be still about a decade away, but by 2030, it might be that the surgeon is really wearing his gloves and just standing and looking at the machines operating. In ophthalmology, we already have a lot of this in terms of different kind of LASIK surgeries and other things where the physician really isn't doing much other than guiding that machine. So we are slowly but surely going towards that. And AI is everywhere, right? It's in our cars, it's in our pockets, it's everywhere. So it is going to come into medicine big time, right? So AI will change the interaction between doctors and patients, but most patients won't even know that it's involved because it's going to be at the back end, but physicians will definitely know. So is this good or bad, right? So I think it's definitely good for the patients and for the community itself, because machines which are well trained will probably make better decisions than physicians, but it may not be so good for physicians. There are pros and cons of these, but the relevance of your diagnostic abilities, to come to a rare kind of diagnosis based on a lot of these clinical features and a lot of investigations is going to go down a bit, because you will have a machine which will actually process all that information and quickly give you that diagnosis or that that form of therapy. And this is a book which is a must-read, I think, for all of us. This is a book written by one of the the greatest minds alive today. He is Yuval Noah Harari. I'm sure you've heard about him and the books as he is written. So this is 21 Lessons for the 21st Century, and what he really talks about to a large extent, right at the beginning of the book, is EdMed. So education and medicine is really going to be revolutionized by this automations in the coming decade, and how EdMed will look like in 2030 cannot even be guessed widely. So how can we be prepared, right? So it's a bit scary that our relevance is going to reduce, but how can we be prepared? AI integration into medicine, it cannot be stopped, right? Because large corporations like Google, Facebook, Apple, and all of these are getting into, all of these. It really cannot be stopped. Almost, at least a lot of diagnostics is going to be automated. A lot of medical fields such as, diabetes care, etc., are going to be automated. So I was recently reviewing a grant proposal from a company or a group of physicians who have formed a company in Israel, and what they're trying to do is automate the way diabetes is managed, right? So physicians are the biggest barriers, is what they show in terms of getting good diabetes control for patients, because patients with the same sort of investigations are treated entirely differently by physicians, and patients with very different kind of features are treated in the same way by physicians. So what they are really showing is that the physician is the main person because of which there is so much variability. So they are trying to bring in automations which may actually sort of undermine physicians. This won't happen overnight. The good news is that this is still evolving, right? And so we can assist this evolution and be a part of it rather than against it. So the main question really is, can I be part of this? What can I do to be associated with all of this? I'm a singleton. I'm a person. I'm just into medical school, or I'm not even out of medical school. How can I be part of this? So I think it's important for you to understand that you can break barriers, right? So this plane is going faster than the speed of sound, and that was, they never thought possible even 4 or 5 decades ago. But man has broken these barriers. So don't think that you can't. You need to find new ways to win, and you need to think. I'm sorry about that. You need to think smart, and thinking smart and working smart, and not just work hard, right? This is a great book. If anyone wants to look at, you can actually also look at this. Right? So how can you be part of this? I think it's important to train yourselves with data. Like I said, don't put all your all your eggs in one basket. So train yourselves with data, learn analytics, statistics, learn to present data well, learn to write well, make an impression. Finally, be good at medical research, because be prepared for the tomorrow today. So I think that is sort of the overall crux of why I was telling you all of these things. So once you know that this is sort of the basis of what I wanted to sort of discuss with you, but now we'll look at how we might get good at this, right? So this I've already shown you before. This is the roadmap to publish. So you plan a study, you execute it, you analyze it, then you write it. Like I said, you need to train yourself. So I actually went ahead and trained myself in the year 2008. So that's 12 years ago. That is me. And this was a highly it was a very intensive training session for 3 days on research methodology. So most crucial aspect, like anything else in life, people say there is this 10,000 hour rule. So if you do something for 10,000 hours, that's a long time, right? But if you do it over and over again for 10,000 hours, you become good at it. So it could be surgery, it could be patient care, it could be reading ECGs, it could be anything, but you have to put in that initial effort. So initially this is a little bit of a recap of what how I went through this. So I did the three-day course at the LICO, that is Lions Institution in Madurai. Then I went to, I was not really satisfied. In 2010, I went to CMC Vellore and did a 1-week training course in all these research techniques. I still wasn't somehow satisfied, and so I went to Wilmera Institute, that is in Johns Hopkins, and I did slightly more than a year's training there. So learning along the way is, I have learned how to do statistics, I have learned how to write papers, and now during my editorship, I'm still learning some of these. So get trained in basic concepts. Trying to publish without training is like really putting a lens inside the eye without putting an incision, or in simple, to give you another example, it's like removing a hernia without an incision, or getting rid of an appendix even without making an incision, right? So it's very difficult to achieve success without training. And nowadays, a lot of technology is available, courses are available easily, you can interact with students, you can choose your teachers, right? So it's really it's within your grasp. So how you can do it, the approach, I've already shown you a bit of opportunities. Now the approach, what we'll discuss today is why I should publish, or why I should do research and then publish, how I should do it, where can I publish based, I'll tell you a little bit about journals, and some take-home messages. So what are medical journals? So I think you all took majority of you took a poll about a couple of hours back, and I saw that a lot of you are already aware of what medical journals are. Believe me, when I was in medical school, I had no idea what a medical journal was, right? So of course it's a long time back, but so you guys are already much ahead of how we were when we were in medical school. So journals are where research is published, right? So when you do all these studies, write a paper, you then send it to a journal, and then they will go over it, and then they will see whether it's the merits and demerits, and then they will publish it. Examples are the New England Journal of Medicine, Lancet, JAMA, British Medical Journal. So these are some of the top journals. This is a hierarchy of study designs, where as you go up this pyramid, the level of evidence improves or getting better. At the bottom is the animal and laboratory studies. Then you have case reports or case series, which are single sort of reports. On the right, if you see, it says there is no formal design to these. Then you look at case control studies, then you look at cohort studies, and then you look at randomized control trials. So these are studies which keep generating better and better evidence, more robust evidence that you can believe as you keep going up the ladder. And finally, when you put all studies together and have a sort of consolidated idea of what literature is saying, that is called a meta-analysis or systematic review. And you really have to go up the ladder as you you have to progress. Initially, it's good to start with case reports. As you go on, get more experience, you should try and do more studies, case controls or cohorts. Later on, you should try and get involved with randomized control trials. And finally, when you have an opportunity, you should probably look at doing some meta-analyses. So I've already told you a little bit about why you should publish, but that is a bit in the future. So what is it that you can get if you publish now? Quickly to summarize, work that remains unpublished in one form or the other is essentially incomplete or undone. Right? So if you do a thesis or if you do a study and you don't tell the world what you found, it's really undone, right? A lot of effort goes waste. And a lot of the thesis, so this is specifically from my field, that is ophthalmology. If you look at a lot of the thesis, they are not published. So a lot of them remains that iceberg really remains below. It remains submerged, and there is lack of novelty. The design is not good. There is not enough training. There is apathy. People are not really aware of these benefits, right? So a lot of the thesis remains unpublished. And if you see from every every subspecialty has an annual meeting. So the All India Ophthalmic Conference happens every year, right? So people present their research in that conference, and based on some of the feedback that they give on the presentations, they actually then go and write the paper and then submit to a journal. But from the All India Ophthalmology Society meeting, only 12% actually go on to get published in a journal. That's that's really ridiculously low. If you look at international meetings, some of them held in America and Europe, the numbers are much higher. So why do you do research, from a current perspective? You can remember the mnemonic FAMOUS, though it does make you a bit famous, but don't take it literally. So the mnemonic F stands for fame and recognition, of course. When meaningful research makes an impact and gains you peer respect, that is what a lot of us are after. Respect from patients, but more importantly, respect from your peers. That really drives you ahead. A stands for patient care, where your pivotal research studies can have potential to enhance patient care, and it can actually influence millions of lives. I've really seen live examples. When I was at Johns Hopkins, that School of Public Health had a tagline. It said, School of Public Health, Bloomberg School of Public Health, saving lives millions at a time. And when I saw that for the first time, I had a hearty laugh, and I said, oh, these Americans, they really talk too much. But as I went through the process and I understood what the school does, for example, one of their studies actually has revolutionized the immunization schedule of whole of Egypt. One of their studies has led to eradication of malaria in many parts of Africa. A lot of their studies have led to the conquering the Ebola epidemic, right? So, actually, they have really saved millions of lives. Then, of course, M stands for make yourself a good clinician. All the deliberations that will happen during your research will make you a better thinker, and you will finally end up being a better physician. O is for policy decisions. A lot of impactful papers, don't think that they are read only by your peers or friends. A lot of the times they may be read by IAS officers, police officers, judges, many people. A lot of news agencies will read your paper. So remember that the paper, once published, will remain on the internet, right? So anybody, even 5 years, 10 years down the line, can actually read it. And a lot of these papers are used to make policy decisions, allocate funds to public welfare projects, etc. I'll show you some examples. Of course, you can get funding and grants, and then personal index. It really means that you start getting better and better. You can really, by doing research, you can go beyond one patient. So this is one example from one of our studies where, the caterpillar, those small insects that you have, they have a lot of hair on their surfaces, and those hair can actually go inside the eye and cause a lot of havoc. So one of our papers actually talked about how this happens and how you can prevent it and how you can actually pick it up before it causes damage. And I have received emails from all across the world, from almost all places where these caterpillar hair causes damage. And so it has actually helped many physicians. I remember one ophthalmologist lady from Italy saying that it has gone into my daughter's eye and I could pick it up and I could treat it because of your paper. So that really made my day. It can help in decision making thought process. So this is one paper, again specific from my field, where you must have heard about phacoemulsification. That is a form of cataract surgery that leads to drop in intraocular pressure. But then people felt that the ultrasound energy is causing drop in the intraocular pressure, but we showed that manual surgery also causes an equal drop, so there probably is no role of ultrasound. So it challenged the thought process that ultrasound energy is responsible for drop in intraocular pressure. And this has really led to a lot of large labs losing a lot of funding because they were looking at ultrasound energy and intraocular pressures. You can actually influence literature if you publish some pivotal papers. So this is one of our papers published almost a decade back now. We don't need to go into nitty-gritties of these. It's a complication after cataract surgery is not very common. So I have reviewed at least 50 papers over the last 10 years on this rare disease. And me and my group, imagine how much influence we might have had on how this whole literature of this has evolved over the last decade. That will be quite large, right? So as an expert in the field, you can actually influence a lot of literature, and that is a lot, that with intense power comes a lot of responsibility, right? So it becomes a responsibility of yours. Like I said, some of these papers can influence policy. So this paper was published recently, and it talked about definitions of blindness, and it talked about definitions of blindness which should be changed. And see, the Hindustan Times has picked up and see the headlines. It says number of blind to come down by 4 million as India set to change blindness definitions. So a simple thing like changing a definition can actually lead to huge change in the overall incidence of a disease, right? So it's apparent then that could happen. But this paper has actually caused a dramatic drop in the funding for the National Prevention and Control of Blindness program by the Government of India. This is another paper which has done something similar. What's in it for me? I think a lot of grants, funding, fame, and recognition, like I've already told you. From a medical graduate point of view, I think this is something very coveted. Is Ivy League residency programs in the US, UK. Many of you might actually be thinking about doing USMLE and having residency programs outside the country. Most of these large residency programs are the Ivy League residency programs, the Yales, the Hopkins, and all of these. They want residents to have published papers before they even entertain your applications, right? So this is one paper which talks about unverifiable publications. But what I wanted to show you is these results. If you say, it says that 2/3 or 75% of residents reported at least one qualifying publication, right? So one publication is a full text or an original article where a lot of patients were involved and other things. So if you really want to get into some of these very top institutions, you must have some publications under your belt. And there is an imbalance where 90% of the world population lives in the developing world, and 90% of papers are published from the developed world, right? So onus is really on all of us to sort of, correct this imbalance. From this, I think it's important to understand that if you go beyond one patient, then you do good clinical research and you must publish meaningful work. So, we talked about why I should do. Now we'll look at some other concepts. We'll look at some nitty-gritties of how you can do this, right? So how, from concept to this publication, how you can do this. I think the first thing is the idea, right? the ideation. So for anything, for a startup company or for for anything that you're doing, the idea is the first thing that comes to your mind. So how do you get ideas to do research? So this is sort of this is something that you can actually, these are infographics which you can find on my Facebook page and website as well. So we'll take a look at some of these. First is you have to zoom into your area of interest or your subspecialty of interest. It could be pediatrics, it could be something as high as urology, or something like general surgery or any other surgical field. And once you home into a couple of areas of interest, you need to look at some of the top journals which are getting published in that field, and you can actually go and subscribe to the e-table of contents. I'll show you an example in ophthalmology, and these are all free, and you can actually subscribe to these e-table of contents. You can the poll we took about a couple of hours back said that a lot of you have heard about PubMed, right? So, which is great. I had really not heard about PubMed during medical school. You can go to PubMed and you can customize PubMed to send you updates from those particular fields that you are interested in. This is an example of JAMA Ophthalmology. This is one of the major journals in ophthalmology. And if you see here, it says get the latest from JAMA. You just put your email and sign up, and it'll keep sending you email. So this is from my own inbox. So it will send you whatever is published in JAMA Ophthalmology in that month. So every month you will get these table of contents. You can actually read some of these papers and see if you might be interested in doing something similar. So I subscribe to many different journals, at least 7 or 8, and once you read these table of contents, remember that your eyes are going through at least 60-70 topics in that particular field, right? So you are bound to get some ideas from this. You can, like I said, you can customize PubMed to send you some particular topics that you might be interested in. We don't have a lot of time to go into the details of PubMed and how to customize it, but you can actually do it. There can be ideas from clinics. So when your clinics start, you may be in your third year or your final year. When you go into clinics, be curious, ask a lot of questions, like I said in my first slide. So unless you ask questions, it's difficult to home in on which is the relevant question to study. So ask questions in your clinics. You can think about joining collaborations. Say at least when you're in your residency, you will look at different groups doing different kinds of studies, and you might say, I want to pitch in, and you can join those groups. These journal clubs are specifically happen more in residency, but they might be happening in all your departments, and you might actually get permission to attend some of these journal clubs where some articles from journals are actually discussed. So these are another good ways of getting ideas, and attend some meetings specifically in your area of interest. Even if you're in your MBBS or training, you can still go and attend some of these meetings, and make sure you brainstorm and have some ideas that you can discuss with some people there, right? So after the idea, this basically, now you have the idea of what you want to do. So now you want to design that study, right? You want to execute it. So how do you design a study? You actually use the PICOT approach, right? So P stands for participants. That really means inclusion, exclusion. That is, whom do you want to study and whom do you want to not study? You need to define these very nicely, right? So you might need help from your seniors, but you must home in on whom, the exact population that you want to study, or the exact nature of the disease that you want to study. I stands for intervention, right? Intervention really means what is the tool that you're using to gather your data. It could be as simple as administering a questionnaire, to as complicated as a cardiac catheterization, or something like that. So the intervention can range from very very simple to very complex, but it has to be well defined, and you have to understand the tool that you're using to collect data is called intervention. C stands for a comparison group. So it could be an active or a passive group, and unless you have a comparison group, it's suppose you're studying something new, it's very difficult to say whether it's better or not from what is existingly being done, right? So it's important, if you don't have this comparison group right at the beginning, and it's very hard to go back and get that comparison group. So think about whether your study will do better with a comparison group. Then O stands for outcome measures. Exactly what are you studying? Say you're studying something like ureteric catheterization. does it improve the output, right? So urinary output. So what is it that you're measuring? Are you going to measure that in milliliters, in liters per day, or whatever that may be? Say in the eye, you're looking at cataract surgery and vision. How are you going to exactly measure that vision? So this is called outcome measure. So it must be defined very well. And T basically stands for type of study. Whether you want to do, like I've already shown you that pyramid, where prospective versus retrospective study, whether you want to do a randomization, what is your sample size, and all of these, right? So T stands for the type of study. So PICOT is the way to design a study. Once you design the study, you want to execute it, right? You actually now want to go into the patient population and actually now collect data, and then you want to analyze it, right? So once you do it, you have data along with you. It is going to be in your Excel sheet or wherever you're storing it. You want to do some statistics from it, right? Once you do statistics, only then will you realize whether what you thought and the idea that you started with, also called a hypothesis, so whether the idea that you had in mind is actually true or not, right? So you need to do some statistics or juice, find the juice from the numbers. So this is a free ebook which is available from my website as well as Facebook page. You can go and download this. It makes it relatively easy. So quickly I wanted to take you through some of these basic terminologies, because you should be aware of this. So everything that you're recording from a patient, say in ophthalmology, you're recording the vision, you're recording the intraocular pressure, you're recording so many different variables. All of these that you're recording they are called variables. In other words, we mean, I'm sure all of you are aware of how an Excel sheet is designed. So every patient has one row of an Excel to themselves, right? That's their sort of their private property now in your study. So every patient's row is different, but the number of columns that you have, they are the variables. So, right, every column in the Excel sheet is a variable. It could be a continuous variable or a categorical variable, where continuous basically means it can vary from 0 to infinity, and it will have a value to it. It will have a unit to it, right? So for example, say millimeters of mercury for blood pressure, right? So or for blood sugar levels, there is a unit to it, and it can really vary from 0 to infinity, something like that. So whenever you're looking at a variable like that, it's a continuous variable. A categorical variable is something like gender. Say it can be either male or female, so there can be only 2 categories, or there might be only 3 categories, right? So that is a example for categorical variable. You might really find it confusing at the moment, but unless you at least hear some of these terminologies, it will be difficult to understand them later. So what we use to find differences in these variables are called tests of significance. So if some of you have gone through preventive and social medicine, and these are generally taught in that, but how we find which tests to be used. So the tests are there on the right. So this is the statistical test. As you will see, it's a student t-test, rank sum test, ANOVA, Krusal-Wallis, chi-square test, and Fisher test, right? So how you actually home into the tests is by asking yourself some basic questions. First thing you have to ask is, what is the type of variable? Is it a continuous variable or is it a categorical variable? Second thing is, you need to ask how many number of groups are there. So if it's a treatment group and a comparison group, there are only 2 groups, but then there may be 2 treatment groups or test groups and 1 comparison group. So there may be more than 2 groups rarely. So this is the second question you need to ask. The third is whether that variable is distributed normally or not. That is whether it follows a bell curve or not. So based on this, you can actually find a test of significance. So we don't have enough time to actually go into details with examples, but this is essentially the overall idea that we use, and we do appropriate statistics. This is again slightly complicated. Different kinds of regressions are there, where it's very difficult to explain, but just keep in mind that regression analysis is probably the best way to determine causation, right? Many times you will find one, say vitamin D deficiency causes headache. If somebody tells you that, immediately your question arises in your mind: is that so? Is it a cause? Is there a cause and effect relationship? Is the vitamin D really a causative factor for a headache? So you will find the answers best when you do regression analysis. There are different kinds of regression analysis called linear regression, logistic regression, and many different ones. Those who are interested can actually read up and get better idea. Quickly, I wanted to tell you something about sample size. Why it is so important is because we want to get closer to the truth. Say you want to do a study on tuberculosis. There are say 10 lakh tuberculosis patients at a given time in India. Now it's impossible to go and test a drug on all the 10 lakhs, right? But then you can't really test it on 10 people, right? You can't say, oh, 5 people got the drug, 5 people didn't get the drug, and this is the final output. The drug is great and dogmatic, and oh, you must use this drug. So 10 is too little, 10 lakh is too large, right? So there has to be a somewhere in between where you will actually get results which will be applicable to the whole population. But then it will still be doable. It will still be, say you studied 2,000 patients or say 1,500 patients, something like that. Then it makes a lot of sense. So the closer your, the number of patients in your study is closer, it can actually be calculated by using certain biostatistical concepts. And so therefore sample size calculation must be done before you start. And how does it influences? It really influences everything. If the sample size is wrong, all your conclusions will be wrong, and subsequently when other papers are going to be published, they are not going to match your studies, right? So that overall credibility may be lost. So, initially you have an idea, then you design the study, like I said, using the FICO approach, you analyze using statistics. Finally, you want to write that paper and publish it in the journal, right? So there are best practices for writing. So that is what I'll quickly tell you now. So the best way forward is to use checklists to write. So this is a great book, again, the Checklist Manifesto, How to Get Things Right. So this is written by medical doctor Atul Gawande. I'm sure a lot of you might have heard about him. If not, I think you should, and you should try and read this book, right? So a checklist approach helps ensure quality, objectivity, thoroughness, and critique, right? So these are all very important concepts when you are sitting to write not just a medical paper, but anything that you're writing, including your theory exams and others. So if you have a checklist in mind, okay, this is how I'm going to write this particular thing, it really helps. Most papers are written using the IMRAD format, where I stands for introduction to your topic that you are actually studying. M stands for methods, that is what did you do. R stands for results, that is what did you find. And D stands for discussion, that really stands for how does what you found, how does it sort of tie into existing knowledge of that particular disease entity. So introduction is what did you do or what was not known, and method stands for what you did, the result stands for what you found, and discussion is for how you can merge what you found into existing literature. So this is a website that all of you should visit. It's called the Equator Network. Equator stands for Enhancing the Quality and Transparency of Health Research. And what this gives is a lot of checklists for you to write different kinds of studies. If you see here, randomized trials, it says consort. So consort is a checklist. These are all freely available to download. Suppose you're writing an observational study, you need to use the stroke checklist. If you're writing case reports, you should download the care checklist and write according to that, right? So these are all mnemonics. Strobe is for observational studies, consort is for randomized trials, record is for retrospective studies, care is for case reports, like I already told you. IMRAD format is used in most studies. This appears complicated, but this is a decision tree which guides guides you to your relevant checklist. Let's take an example. So first question on the top left is, was the research on humans? Answer is most likely yes. Did your research generate quantitative data? So if you've done, say, a study on 300 patients or so, you would have generated a lot of data, that is quantitative data. You say yes. The next question on the below that yes is, did you combine and analyze results from previous studies? Not really, right? So this is data only from your study. So you say no. The next question is, what was your study a randomized trial comparing two or more health interventions? So if you're doing if you say you're doing a prospective or an observational study where there is no randomization as such, again the answer remains no. And then the question is, did your study explore the relationship between exposure to risk or protective factors and outcomes? If the answer is yes, you look at the stroke checklist, right? So this is the commonest checklist that we use for writing, because most of us will be doing a prospective or a retrospective study. So look at that. That's a 21-point questionnaire. It's like questionnaire. You write answers to a couple of lines to each question based on what you have done, and then your whole manuscript is ready, right? So you should use checklists to write. At least you will avoid some non-negotiables, right? So some very crucial elements of a paper may actually get missed if you don't use these checklists. So these are all mnemonics. Like I said, the STROPE checklist. STROPE stands for Strengthening the Reporting of Observational Studies in Epidemiology Initiative, right? So RECORD stands for reporting the studies conducted using observational, routinely collected health data. That's the record statement, CONSORT. So all of these are, these are all mnemonics. So you can actually go to explore it, and you will have, a lot of knowledge about how to write papers. Other applications of checklists, basically, in addition to writing your papers, these checklists can be used for departmental journal clubs. Whenever you're reading a paper, even when you're done with your medical school and then you're actually practicing, many papers are going to get published, right? So some will be good, some will not be good, and some will compel you to change your practice patterns to improve some aspects of patient care. But should you do it or not depends really on how well the paper was done, right? How well the study was done. So unless you're aware of some of these aspects, you will really not be able to tell which paper is good and which paper is bad, which is really happening today, on a very large scale. While government of India is, and ICMR is also giving out guidelines for management of COVID. A lot of them are not evidence-based. A lot of them, like HCQ for COVID or so many of these others, even see these durations between the first and the second dose of vaccines and all. I don't know what they're doing, but the guidelines are extremely clear if they had referred to proper papers. But then, unfortunately, if you don't refer to proper papers, you're going to get into a situation that we are in today, right? So these are some of the other applications of checklists. This is another website that most medical journal editors, so I am on the editorial board and I look at these websites. These are called the International Committee of Medical Journal Editors, and this also gives you guidelines on how to write papers. This is one of my papers in Indian Journal of Ophthalmology in 2014, and this is free to download and has been downloaded more than 4,000 times. It's called Publish or Perish: The Art of Scientific Writing. So if you find these CONSORT and these other checklists quite difficult to follow, so this is a good paper for starters that you can use to write some papers. This is my website. It's called Sengupta Research Academy, and it has got some training courses for you to understand how to do research. It also has some free lectures. Please explore them. These are some of the modules which are there, where there is one free module which talks about introductions and why you should do research. Is something that I've already told you. It has a bit of my own story as to how I went through my MBBS, and then what was my first project and how I got along with it. Then there is a module on literature review, which talks about how to choose a research topic, which we have briefly discussed. There are many more details in these lectures. PubMed search and how you can do it the best way, what are the different study designs, and all of these, these are all bite-sized lectures, 8 or 9 minutes. You can actually learn a lot from some of these. Then there is a whole module on biostatistics. It teaches you how to make an Excel sheet. That's one of those, one of the lectures. And believe me, most physicians do not know how to make Excel sheets. So, that is true for 90% of physicians all over the world, because I have analyzed data from people in Australia, from people in Europe. I got an Excel sheet from 700 case series in Italy, and imagine it was in Word document. It was not even in Excel, it was in Word. So, so it was impossible. I had to, translate everything into Italian and then send it back to them and get them to make an Excel sheet. And this is a lecture which I had sent to them, telling them, please use these guidelines and make a good Excel sheet. So it's not very common to make a good Excel sheet for data analysis. So learn some of these things. Then, like I've already told you a little bit about manuscript writing. What we have discussed a little bit today is the checklist-based approach. But there are different nuances of how you can choose a journal, how you can write original articles, case reports, which are a good way to start. And then what a reviewer wants, and then what are the common causes of rejections of papers, which are a big setback especially when you're starting. And if you have 2 or 3 rejections, you will not want to go ahead and do more. So it's important to not make those mistakes. So from an editor's point of view, I can tell you what are the common mistakes which people make. So these are something that you will learn from these lectures. There are other interesting things on the website. There are many blogs, some on p-value, some on regression analysis that I was briefly referring to. There are many other blogs on, how to convert your thesis to a journal article, important sample size, something which you may be interested in is planning a study. So please make a point to go through some of these blogs. There are about 35, 36 blogs. I think you'll find some of these interesting. And there are more than 3,000 students which are enrolled in these lectures from 79 countries today. So I'm quite proud of my team and how much we have able to get through to people in different countries, and so it's making quite a difference to a lot of people, and a lot of MBBS students are already enrolled in this, right? So final take-home message, I think, is you must train yourself. You then design and conduct a study, you analyze the data, you write your manuscript, and then you finally go ahead and publish, right? So like I had said in the beginning, you must think about breaking barriers, be future ready. I've already given you a quick sort of overview of how practice might be in 2030, and I think you should be ready for the future and contribute to that rather than oppose it. So thank you very much, and I'm quite happy to take questions if you have any.
Thank you so much, sir, for explaining clinical research so well. And I'm sure everyone here is really motivated and looking forward to pursue that now. I would now like to hand over to our PRC Director, Reena Mehta, for the question and answer session.
Good evening, sir. It was a lovely seminar. It was really, really lovely. I have learned a lot about research. You covered like a lot of questions that we got in, but just like a few, I would say to ask you, what do you think would be our approach, our thought process as a medical undergraduate or even postgraduate student while starting with the research, while just in the process of selecting a topic?
So, like I already told you, how you can select some good topics. First and foremost, you should understand your own limitations as well. It's not good enough to say I want the star, because it may really be out of reach for you. So look at your institution, look at where you are already situated, and what is doable, what is feasible rather for your own settings. Second thing is then you must look at what are the common diseases which are there in that department. Say you're interested in gynecology research. Now, what are the commonest conditions that you're seeing? So you're likely to have a larger sample if you are looking at only those conditions, right? So look at your limitations, look at the number of patients that are there in larger numbers in that particular subspecialty, and now you're already zooming in or honing into something that you can do, right? Now, once you do that, then you do a proper literature review, right? To look at what has not been done. So most people will do a literature review and say, "Arey yaar, sab kuch ho chuka hai. What more can I do?" But then really you need to find what has not been done. And what has not been done can only happen once you start asking questions to yourself. So when I used to read, even during residency, if I'm opening a book chapter and reading, I would have already seen a couple of patients say here and there, and somebody would have uttered something in my head during that period. So I would really jot down what I know about that. Then I would jot down what I want to know about that. So the questions, and then I would try and find answers to these questions in that text. Right? Because at least 30-40% of that text I would have already heard from somewhere here and there. So, say if I have 9 questions to answer, say, what happens to this patient after 1 year? Or what happens to this patient on the long term? I'm doing a catheterization or I'm doing a angiography. Does it really last for 2 years, 5 years? What really happens? So when you have some questions, you will find that you're finding answers to, say, 5 out of those from your textbook. Out of another 5, you might find 3 answers on PubMed, but then 2 questions will still remain unanswered. Now, when you have that, that is a question which is worth answering, right? That is a question which is worth pursuing, collecting data, finding answers to that question. So, that is, I think, how you can go ahead, where you first look at your own limitations, then the disease process that you can study based on the number of patients that you have, then do a literature review, and then zoom in on what you can really do.
Also, so that was a lovely answer again. Like, no one has explained it that easily to us. Also, sir, what would you think is the importance of having a mentor during the research, and how would we select the right mentor for us?
I think that's a really important question. Again, so good questions. I'm happy that all of you are asking questions. Going back to my first slide, unless you ask questions, in our education system, questions are forbidden, right? Most of us will be, they will run behind you with a broom saying, "Oh, you asked too many questions." But then I think that is the sort of change in mindset which is really required. A good mentor will allow you to ask questions. I think that is the first and foremost of how you identify is, they'll never be angry at the kind of question that you ask. Even if they don't know, they'll say it. They'll say, "I don't know. Why don't you find out?" Or "Let's sit together and find out." So that is some, one way of where you can identify a mentor. Selecting a bad mentor will set you back almost a decade. So, don't select the wrong mentor. So the way you select right mentors, like I said, is one is, they will be patient with you. They will try and answer most of your questions. They would be, have a relatively good practices. They would have a good, sound scientific approach. Whenever you are saying rounds or you are looking at some, patients and their approach, you will find that it is scientific, right? They won't say, oh, do this. They'll say, do this because this, right? So because there is evidence saying that, say this large randomized trial, say, they will say that the ACTT trial said that remdesivir works in this situations, and it doesn't work in these situations, and that's why I'm using it here. Right? So it'll be, I don't think that's very hard to identify somebody who has that approach, right? So identify somebody who has a sound scientific approach, who is probably reading papers, right? Who is citing papers as you go along, right? That's not very often that you will find somebody who will, at the top of their head, cite a paper and say, oh, this paper said this paper said this. That means somebody who is very actively reading, very involved in what they're doing, passionate about these things, and then allows you to ask questions. So I think that is the sort of hallmark of, I've seen so many of these when I was at Johns Hopkins, and I did not see too many of these when I was in India. So, but you can still find some good ones if you look close.
Okay, sir. So the participants have a few questions. There's a question by Sakshi. She's submitting a case report to the British Medical Journal, but they need a cover letter to the editor, and she wants to know what all would you usually involve in like a cover letter? What would you include in it? Whether there should be personal details or just your research?
So, a cover letter for a relatively large study is different from a cover letter which is for a case report. So case report is just one, it's one single case, right? So a cover letter basically, from an editor's point of view, when I'm reading a cover letter, I want to know whether I should send this paper out to a peer review or not, right? Is it interesting enough for my readership later on? So your uniqueness and your education value of that case report should be part of that cover letter. It should be only 2 lines. Dear editor, it gives us immense pleasure to submit this to your esteemed journal. Then you say, this is the unique feature of this case report, and this is the educational value of this case report. Not in those terms, but you say, this case is rare because, XYZ. So that really shows you the uniqueness. Then you say, this was managed in this challenging management, and it went like this. Now that is the educational value, right? So these are the couple of things that an editor will look at for when they are reading a cover letter for a case report. From a original article, first you have to tell what has not been done. So in one sentence you say, this is a common disease, but surprisingly this has never been done. So that suddenly piques the interest, right? The editor will say, oh, really? Has it not been done? Oh, I'm not so sure, but okay, fine. Now, let my reviewers take a look, and then it will go for peer review. Believe me, at least, say top journals like the Lancet or the BMJ or any JM have about 70% rejection rates at the editorial level. It will never make it to peer review at all. That's because, a lot of this has to do with how the cover letters are written. They're not, the editor doesn't have time to go through all the submissions and the entire nitty-gritties of the paper. They'll look at the cover letter, they'll say, oh, this makes a lot of sense. Then they'll look at the abstract of the paper, which is the précis, right? So that's like a 250 word document. That's like an executive summary, or, once the cover letter makes sense, they'll go to the abstract. Once the abstract makes sense, it will go to peer review. If the cover letter doesn't make sense, it will never go ahead to the peer review at all, or they will not even probably read the abstract. So it's important to therefore construct that, like I said, uniqueness and the uniqueness and your educational value are the 2 things that you should include in your cover letter.
Okay, so so we have another question by Supriya. She's asking how to cut down the number of literature reviews since it's very vast and confusing to read.
Right. So I think you need to do a proper training on literature review, because PubMed search box is not as intuitive as the Google search box, right? Google search box really is AI driven. There is a lot of artificial intelligence which is going at the back end, and you don't even realize. Similarly, a lot of AI goes behind how medical diagnostics will work in the future. PubMed doesn't work like that. It is, at least as of now, it is not AI driven. So you can't just type whatever you want. So you need to use keywords. Right? Keywords are the most important way of getting to the right search that you want. And in short, you need to match your keywords that you have in mind to the mesh terms which are there. So first thing is you need to use proper mesh terms, which are which are determined by PubMed. Once you use mesh terms, then you can use something called the advanced search engine, where there are Boolean search operators. A lot of this is there in the lecture series that I showed you just now on PubMed overview and PubMed search. There are a lot of differences between PubMed Central, Medline, and PubMed, and how you can actually cut down. So there is a nice example that I have given in that, where say you're looking at incidence of post-cataract surgery endophthalmitis, which is like an infection in India, right? So initially you look at post-cataract endophthalmitis, then you reduce it to endophthalmitis in India, then you go down or zoom into post-cataract endophthalmitis, and then you only look at incidence, because there are many aspects of a disease: incidence, risk factors, outcomes, and so many other things, right? So you just then type incidence. So you will see that the search goes from 1000 to almost 35 or 36. That is really what you're looking to do. I think that's what your question is about. So you need to understand how to do a good PubMed search, and for that you need to have a little bit of training in how that is done. It's not really hard at all.
Okay, sir. So we'll be taking the last question for the day. The question is whether it's important to be a clinical or to be a research assistant to like get into learning about research and publication, just like how you have to do a shadowing or an observership in clinics.
See, that's what. So it really depends on where you are. If you're in India, you can, also depends a bit on your mentor and how they are willing to mentor you. Some, especially in India, there is, not too many will handhold you, but they'll say, "Acha, tum karo, main hu." It's going to be like that. So if you know how to do it, you can do so many things in India. But if you're outside India, you really have to go through all these rigors. You have to do an MPH, and then you have to go through a lot of red tape to get your hands on patient data. Like for example, when I reached the US, it's Johns Hopkins. That's like the biggest institution. That's the one which is keeping tabs on all the COVID numbers. You would have seen, right? So I know a lot of those people who are actually doing that, and a lot of them are from the from the medical background, of course. So they made me do a lot of online courses. There is HIPAA compliance and so many different things, and only then you are eligible to even look at some patient data. So it's not easy to get access to patient data when you're outside India. But here, it's all over the place, right? It's lying on the ground. One paper would have been flying loose, and then all the major oncology treatment would be on that sheet, and it's just lying loose on the ground. It's possible, So there's a lot of data flying over in India. People don't care about data, but data is the currency of the future, right? So it's a lot of it is there here. So only thing is you should know how to use it. So get trained, know how to use it, and you can do so many more things if you are here. So like I said, so if some of you are interested in USMLE and other things, it's easier to get publications when you are in India. During your third year, you initiate it. You do a couple of case reports, then you do maybe a couple of full original articles. So by the time you are finished with your internship, you may have 4 or 5 under your belt, which is a very large number. Hardly any Americans would have that much at your age.
Okay, so thank you for your explanation. It was lovely having you. It was an honor to have you. And research is made very easy for us. Like now we kind of understand a major part of it, and the remaining I'm pretty sure everyone's going to be visiting your website and like learning more from all the lectures. So thank you for conducting this.
Super. Thanks so much. Yeah, it's always nice, like I said in the beginning, to interact with young and impressionable minds, and this is where trends are going. So what I tried and showed you are trends. These are going to happen as we progress. So it's important to notice those trends because you are still young, right? So it's very hard for you to see those trends. So that's why I thought I'll show you some trends, and this is where it's going to be. So you can be prepared. You can ace it if you know it, right? So all the best to all of you, and I hope a lot of you will get into the nitty-gritties of these. It's not very hard. It's a very methodical process, and once you ace it, people will run behind you.
Thank you, sir.
All right.